Ion exchange chromatography, IEX or IEC, is a liquid chromatographic separation technique in which ionic analytes are reversibly retained by a stationary phase carrying immobilized charged functional groups. Separation arises from differences in the strength of electrostatic interactions between analytes and the stationary phase. In cation exchange chromatography, the stationary phase is negatively charged and retains positively charged species. In anion exchange chromatography, the stationary phase is positively charged and retains anions. Retention can be controlled principally through pH, ionic strength, counterion composition, and gradient conditions. The underlying ion exchange resin is not consumed chemically during normal operation. Rather, it provides an insoluble polymeric matrix containing fixed ionic sites and mobile counterions. The user's supplied text describes these resins as cross-linked three-dimensional polymers, most commonly based on styrene divinylbenzene, PSDVB, with functional groups such as sulfonic acid and quaternary ammonium groups introduced after polymer formation. IEX is important in analytical chemistry, protein purification, water treatment, pharmaceutical processing, metal ion separation, environmental analysis, and modern ion chromatography. Historical development. The major turning point in IEX came in 1935, when Basil Adams and Ernest Holmes demonstrated synthetic organic ion exchange materials based on phenol formaldehyde chemistry. An amusing historical detail is that their discovery was connected with experiments showing that crushed phonograph records exhibited ion exchange properties. During and after World War II, ion exchange became an important tool in nuclear chemistry. Synthetic resins enabled difficult separations of rare earth elements and radioactive fission products, including work associated with the Manhattan Project. Later, ion exchange chromatography became a major method for biochemical separations. Amino acid analysis and protein purification were among its early landmark applications. Modern ion chromatography emerged in the 1970s. Structure and chemistry of an ion exchange resin. A conventional synthetic resin can be thought of as the addition of a functional group and counterion to a cross-linked polymer matrix. Today, styrene divinylbenzene, PSDVB, is the dominant synthetic matrix. Styrene provides one vinyl group, whereas DVB contains two vinyl groups, and therefore cross-links the growing polymer network. Suspension polymerization in water produces approximately spherical beads. The amount of DVB controls cross-link density. Higher DVB, more rigid, highly cross-linked matrix, lower swelling and slower ion diffusion. Lower DVB, greater swelling, higher water content, and faster diffusion, but reduced mechanical strength. A conventional PSDVB resin often has a cross-linkage of roughly 8% DVB, although commercial materials span a much wider range depending on their intended application. Two broad morphological classes are important. Gel-type resins contain a relatively homogeneous polymer network. Their pores are largely generated or enlarged through swelling and functionalization. Macroporous resins are manufactured with a permanent pore structure and therefore provide greater accessible surface area, improved mass transfer, and greater resistance to certain chemical and physical stresses. Modern chromatographic media can also use agarose, cellulose, acrylic/methacrylate polymers, silica polymer hybrids, and other matrices, particularly when protein biocompatibility, mechanical strength, or high-pressure operation is important. Preparation of resin beads. Polymerization. Historically, polymer bead production is by suspension polymerization in a stirred reactor. For a classical PS-DVB resin, styrene and DVB are dispersed in an aqueous phase containing suitable suspension stabilizers. An organic monomer mixture in an aqueous solution forms tiny, spherical droplets. These organic droplets become the precursors to the final resin particles. An oil-soluble radical initiator polymerizes the vinyl groups inside the dispersed organic droplets. Styrene plus DVB yields cross-linked PS-DVB beads. The suspension is heated to trigger polymerization, turning the liquid droplets into hard, insoluble plastic beads. Control of agitation, droplet size, monomer ratio, initiator concentration, temperature and suspension conditions determines the bead diameter and particle size distribution. These resin beads are best described as like a Swiss cheese. The holes/pores are produced by introducing a porogen, which gives the resin other improved characteristics for certain applications. Typical resin particles are on the order of 300 to 1,200 micrometers, while other commercial formats can be substantially smaller for analytical HPLC applications. Porosity and swelling. Following polymerization, the initially hydrophobic PS-DVB matrix has little water uptake. Functionalization introduces ionic groups that strongly hydrate. The resulting osmotic and hydration forces cause the polymer network to swell until balanced by the elastic restoring forces of the cross-linked network. Thus, resin performance reflects a compromise between mechanical strength and swelling and pore accessibility and ion diffusion and exchange capacity. This relationship is particularly important in chromatographic separations because analytes must diffuse into accessible exchange sites rather than merely interact with the external bead surface. Attachment of functional groups. The polymer matrix is chemically modified after bead formation to introduce ion exchange sites. Cation exchange resins. A classical strong acid cation exchanger is prepared by sulfonation of PS-DVB, producing covalently attached sulfonic acid groups. In aqueous solution, the sulfonate group remains immobilized on the polymer, while H+ is the mobile counterion. Common cation exchangers include strong cation exchanger, SCX, sulfonate or sulfopropyl groups. Ionization is maintained over a broad pH range. Weak cation exchanger, WCX, carboxylate/carboxymethyl groups. Charge depends more strongly on pH. Sulfonate/sulfopropyl and carboxymethyl chemistries are representative strong and weak cation exchange groups. Anion exchange resins. Anion exchangers require positively charged functional groups. A common industrial route is PS-DVB to chloromethylated, PS-DVB to amination to quaternary ammonium resin. For example, treatment with trimethylamine produces a strong base quaternary ammonium exchanger. Common classes are strong anion exchanger, SAX, quaternary ammonium groups. Weak anion exchanger, WAX, protonatable amine groups whose charge depends on pH. The preparation of anion exchange resins is chemically more involved because chloromethylation is commonly followed by substitution with an appropriate amine. Specialty chelating resins incorporate ligands such as aminophosphonic, thiol, or other donor groups. These materials exploit both ion exchange and coordination chemistry to selectively capture metal ions. How ion exchange chromatography works. A packed IEX column contains resin beads equilibrated with a buffer. The analyte mixture is introduced under conditions where some components possess a charge opposite to that of the stationary phase. For example, consider a cation exchanger. A divalent cation can displace a sodium ion because of stronger electrostatic interaction with the fixed sulfonate groups. For an anion exchanger, different analytes have different affinities for the stationary phase. Consequently, they migrate through the column at different rates and dilute as separate peaks. For biomolecules, retention depends strongly on the net charge and charge distribution of the molecule at the selected pH. Proteins are particularly useful examples. At a pH below their isoelectric point, IBPI, they tend to be positively charged and can bind to cation exchangers. Above their PEI, they tend to be negatively charged and can bind to anion exchangers. Elution is commonly achieved by increasing ionic strength, usually with NaCl, allowing salt ions to compete for exchange sites, changing pH, altering the ionization state and therefore the charge of the analyte, changing counterion composition, or using specialized displacement conditions. Modern IEX separations therefore depend on careful control of pH, conductivity, temperature, flow rate, and stationary phase capacity. Major applications. IEX has an unusually broad analytical and industrial scope. Biochemistry and biotechnology, protein purification, monoclonal antibodies, peptides, amino acids, nucleic acids, and charge variant analysis. Analytical chemistry, inorganic anions and cations, organic acids, pharmaceutical counterions, and trace ionic contaminants. Water treatment, removal of calcium and magnesium ions for softening, nitrate, sulfate, arsenate, perchlorate, and other ionic contaminants. Metallurgy and environmental chemistry, selective recovery or removal of metal ions and radionuclides. Ion chromatography, determination of trace ionic species using conductivity. UV, electrochemical or mass spectrometric detection. IEX coupled with MS has become increasingly useful for complex environmental, biological, and pharmaceutical samples. Resin regeneration. Eventually, exchange sites become occupied and the resin reaches exhaustion. Regeneration reverses the loading process by exposing the resin to a high concentration of the desired counterion. Each resin/application has its typical regenerant. Sodium cation exchanger, regenerant NaCl/brine. Proton cation exchanger, regenerant mineral acid. Chloride strong anion exchanger, regenerant sodium chloride. Hydroxy anion exchanger, regenerant sodium hydroxide. A typical cycle is service/loading to backwash to regeneration to slow rinse to fast rinse to re-equilibration to service. Regeneration does not destroy the resin. It converts the resin back toward its original ionic form. The spent regenerant, however, contains concentrated contaminants and must be treated appropriately. Resin fouling and its control. Fouling is the loss of effective exchange capacity or mass transfer performance caused by contaminants that block pores, occupy exchange sites, or interact irreversibly with the polymer. Important foulants include suspended solids and colloids, iron and manganese oxides, silica, oils and greases, natural organic matter, proteins and other macromolecules, bacteria, and algae. Typical symptoms are increasing back pressure, longer rinse requirements, altered breakthrough curves, declining capacity, changes in selectivity, and deterioration of effluent or chromatographic peak quality. Fouling control should begin with pre-treatment, rather than aggressive cleaning after the resin has already been damaged. Filtration, clarification, activated carbon treatment, and appropriate pH adjustment can substantially reduce the contaminant burden. Cleaning chemistry must be matched to resin chemistry. Depending on the resin and foulant, acid, caustic, reducing agents, salt solutions, or carefully selected surfactants may be used. Excessively aggressive treatment can hydrolyze or otherwise damage functional groups and shorten resin lifetime. Disposal and environmental considerations. An important distinction is between spent regenerant and spent resin. Regeneration transfers contaminants from the resin into a concentrated liquid waste stream. Replacing the resin transfers contaminants into a solid waste. Neither should automatically be regarded as ordinary laboratory or municipal waste. The correct disposal route depends primarily on what the resin has captured. A resin used for routine water softening may have a very different regulatory profile from resin loaded with chromium, arsenic, PFAS, radioactive isotopes, toxic metals, or hazardous organic compounds. EPA guidance specifically notes that exhausted resins containing removed contaminants may require special hazardous waste handling. For laboratory and industrial operations, the appropriate procedure is therefore to identify the contaminants loaded onto the resin, determine whether the spent resin is hazardous under applicable federal, state, and local regulations. Do not discharge concentrated regenerant directly to a drain unless specifically authorized. Collect contaminated regenerant separately and characterize it before disposal. Use an approved hazardous waste contractor when required. Document resin identity, operating history, and contaminants captured, particularly for metal, radionuclide, or PFAS-loaded materials. For radioactive applications, additional controls are required because regeneration may produce a highly concentrated radioactive liquid, while the spent resin itself can remain radioactive. EPA documentation describes treatment, controlled storage, or disposal as appropriate management pathways. Concluding perspective. Ion exchange chromatography is fundamentally a reversible electrostatic separation process, but its analytical performance is determined by sophisticated polymer chemistry. The resin must simultaneously provide high exchange capacity, controlled swelling, accessible porosity, adequate mass transfer, chemical stability, and mechanical strength. PS-DVB remains a particularly important platform because its cross-link density and post-polymerization functionalization can be independently manipulated to tune these properties. From a materials science perspective, the central design problem remains the same. Optimize the relationship among fixed charge density, cross-linking, pore architecture, swelling, ion diffusion, selectivity, and chemical stability. Proper regeneration and pretreatment then determine whether that engineered material can retain its performance over hundreds or thousands of operating cycles. This combination of polymer chemistry, transport phenomena, equilibrium thermodynamics, and analytical selectivity is what makes IEX both a classical and still highly relevant separation technology.